Okay, thank you, and I'd like to thank the organizers for this opportunity to speak about my work, talking about the genome-wide interplay between chromatin states, transcription factor binding, and regulatory motifs across 6 human cell types. So the basis from this work started looking at integrative chromatin state modeling as part of the ENCODE consortium efforts. So if you heard Joe Decker's talk yesterday, he mentioned a 7-state chromatin state model. And the basis for that was actually the integration of 2 more detailed chromatin state models, one from Bill Noble's group and one that I had learned based on 14 different inputs using a set of histone modifications as well as CDCF and some DNase hypersensitivity open chromatin assays across 6 human cell types. And we used the method chromHMM, which is a published method that I've presented at this meeting in previous years. And in contrast to previous models where we had focused primarily on the histone modifications, for the first time we included these open chromatin assays into our modeling. And we had learned a common set of state definitions across 6 cell types, but the state assignments could be varying across the cell types. And I'd like to point out a few of the specific states that I'll be referring to during this talk. The state here, which is associated with active promoter regions. And I've boxed in this showing that there's a lot of states which are associated with open chromatin but have different histone modifications. So in this figure, each row represents a state of our hidden Markov model, and each column one of the assays. So you can see there's different states all associated with high levels of open chromatin. So the numbers here indicate what percentage of the locations in that state have that mark considered present. And we have another state here which is associated with active enhancer regions, which have a different combination probabilistically of histone modifications. As well as, interestingly, states which are associated with DNase hypersensitivity but have very weak or limited histone modification signal, as well as distal CDCF potential insulator sites that are associated with open chromatin. And then, actually, a large part of the genome is what we call quiescent states or low signal states, which have limited or no detection of any of the assays which we were looking at in this study. So one of the benefits of the working with the ENCODE consortium is in these matched cell types to which we have these chromatin data, there's also a large panel of transcription factors mapped in the cell type. So in the last ENCODE analysis round, there was, at the time, 119 regulators mapped, in total of 456 experiments. And 321 of those experiments were in matched chromatin states in cell types. And we were interested in now taking these regulators and sort of systematically understanding their chromatin state preference. And because we had our chromatin states defined previously, we could go for each experiment and take the locations where the transcription factor binds and see if there's a relative preference for specific chromatin states. So what you're seeing in this figure is a subset of the regulators that were mapped and a subset of the experiments, color-coded by the cell type. And each row is row normalized, where the yellow indicates this chromatin state which had the greatest enrichment for binding peaks of that regulator. And it turns out that most of the states which have the highest enrichment were one of these open chromatin states, such as the active promoter, the active enhancer, or this open chromatin state with CDCF. There are also some exceptions to the general trend. There was some regulators, SetDB and TRIM28, which had the highest preference for a state which was associated with DNase hypersensitivity, but limited active histone modifications. And REST had a general preference for a very different set of transcription factors. And these preferences were reproducible for these factors across different cell types and sometimes experimental perturbations. So this is showing these patterns of the regulator preference divided into 10 clusters based on a k-means clustering of all the experiments in their matched cell type, looking at over 300 transcription factor binding experiments. And you can see that there's a large class of experiments which had a general preference for promoter regions or enhancer regions, or a balance between them, as well as the insulator regions, the SetDB and TRIM28, which I mentioned on the previous slide, and the rest NRSF transcription factor. So that was all looking at matched cell types, where we were taking the regulator's binding and comparing it to the chromatin states that the transcription factor was mapped in. We next asked, if we take the locations which the transcription factor binds and compare it to the chromatin state maps, both in the cell type in which the regulator is bind and also, mapped, and also other cell types which might not match the experiment. So what we did here, what you're seeing here is a subset of the cell types where each larger column represents one of the states, and each subcolumn the relative enrichment in the specific state. So when you see something like this, this indicates fairly cell type specific preference for enhancers only in one specific cell type, while this means it has a relatively consistent preference for a promoter region. So this now gave us over 60,000 data points of the combinations of chromatin states, the regulator experiments across the cell types. So we had a computational method to order these experiments in an unbiased way to get an unsupervised view of the complexity of the ENCODE data in terms of the intersection between all chromatin states and transcription factor binding. Computationally, we tried to minimize the distance between neighboring rows. And you can see here these general features, such as the promoter state is constitutively enriched for the regulators, and as well as experiments associated with this CDCF insulator state. While the enhancer states can have very cell type specific enhancer enrichments matching the cell type in which the regulator experiment was conducted. And then in other cases, experiments were automatically ordered together, which all had a common regulator. So in this case, 9 of the 10 REST experiments were all ordered together, and they had a consistent chromatin state preference across the experiments, even though they were conducted in different cell types. So that was all still considering one regulator across multiple cell types. In a number of cases, a regulator was mapped, the same regulator was mapped consistently in multiple cell types. So we asked the question, how does the binding states differ if, for a regulator which is mapped in multiple cell types, if we divided the locations bound into places which were uniquely bound? In this case, we were using GM12878 as our reference cell type, and focusing on regulators which were mapped in GM12878 and at least one of these other cell types, which were shared between GM12878 and one of these other cell types, and those places which were excluded, only bound in one of these cell types, but not one or more of these cell types, but not GM12878. And you can see that the promoter states had a shared highest enrichment for the shared states, while the uniquely bound locations were preferred for the enhancer states. And the locations which were associated with open chromatin without the active histone modifications, as well as a more repressive K27ME3 associated state, had a preference for excluded binding, places which were bound in another cell type, but not specifically in GM12878. And we also were interested in the question about how the motif usage varied among the chromatin states which we were looking at for the transcription factor bindings, as well as within peaks. So we asked the question for each of the regulator we were looking at and a known motif for it, whether we saw enrichment for the motif for the regulator in each of the chromatin states, or if we saw significant depletion. And we found what you would expect, that the states for which we saw open chromatin and general transcription factor binding enrichment tended to have an enrichment for motifs. And I'm going to highlight 3 states here to contrast with what I'm going to show in the next slide. So the active enhancer state, a state associated with open chromatin with limited active histone modifications, both had an enrichment for motifs within the state, while there was a depletion in this quiescent state, which was lacking any active signals. But next, we conditioned specifically on locations in which there was a peak. And we were asking the question, are we more likely to have a regulatory motif within peaks in certain chromatin states than others? And it turns out that the situation of the motif usage becomes different in that we still see enrichment for this open chromatin state lacking active histone modification peaks to have a motif relative to the baseline motif enrichment within peaks. But also, if there's a peak in one of these quiescent or states, it tends to more likely have a motif. It's more likely to be motif-driven. While if we have a binding peak in an enhancer, it's less likely to have a motif in one of the active enhancer regions. And the last thing we looked at was the co-binding of transcription factors in the context of these chromatin states. So in this figure, we first looked at the direct raw enrichment of the pairwise enrichment of transcription factor binding. And as you can see, this is showing the pairwise fold enrichment on a scale up to 500-fold. There's actually high enrichment, and there's a lot of structure to this enrichment. And it turns out we've annotated it here. The experiments, this is all in HepG2, and the experiments in this grid were computationally ordered in an unbiased way. And you can see the structure, and then we labeled each row by the cluster that experiment was assigned to in that original clustering I showed, and we've color-coded that based on the clustering the cluster was assigned to. And you see that these clusters all here were assigned to the same chromatin state preference, while this block also had a similar chromatin state preference. So this led us to ask the question, what happens if we condition on the factor's chromatin state preference? What happens then to this pairwise enrichment matrix? So this is along the lines of what Saurabh mentioned in Fly, but we're going beyond just conditioning on DNA accessibility, and we're using the full chromatin state information, including DNase and histone modifications. And you can see that now our scale of enrichment goes down to 50-fold, and pretty much all the structure that we observed previously is lost and can be explained by the chromatin state preferences of each factor individually. So what we've done here is we've taken each factor's individual pairwise enrichment, and we've conditioned what we would expect the joint chromatin state enrichment is and show what it looks like now. But there are some exceptions. This is in K562, where this is before conditioning and this is after. And in some cases, you still see some blocks of co-enrichment which are maintained after conditioning. And sometimes they have very clear biological interpretation. These pairwise experiments are all associated with POL3 associated factors. And these are a set of heterochromatin-associated factors whose pairwise enrichment goes beyond what you would expect by their individual chromatin state preference. This isn't necessarily all to say that the pairwise enrichment, all the pairwise binding is because of the shared chromatin state preference. It doesn't exclude the possibility that two factors could co-bind and establish a chromatin state, but it does raise the question about the possibility that a lot of co-binding could be explained by shared chromatin state preference. So just to summarize, I showed the diversity of open chromatin states through our integrative chromatin state modeling and how that could be used to systematically categorize the chromatin state preference of regulators, and showed how the motif usage can be viewed differently in terms of the chromatin state preferences, and that the co-binding follows the chromatin state preference. And I'd like to acknowledge Manolis Kellis, who was my postdoctoral advisor, and this work was joint with him, the ENCODE consortium, who generated all the data which we were looking at, and also mentioned specifically Puya Kradpur, who generated the motif instances through the ENCODE consortium, and Anshul Kunaji, who led the peak calling, which played an integral part of this analysis. And thank you for your attention.
We have time for one question. If you want to go get some. Please.
So the enhancers were, in general, enriched for motifs, but once you condition on being in a peak, they're less likely to have a motif compared to other states. I'm not sure I understood the question.
To the other, the hypersensitivity of the piece, is it same across both conditions, is my question.
So when you're comparing two transcription factors, and I mean the same transcription factor in two cell types, is the DNA's hypersensitivity the same associated with them?
No, well, I'll ask you.
Okay.
Okay. So